Okay, good afternoon. It's really a great pleasure to be here. First, I want to thank the organizers for giving me this opportunity to be here and to participate in this really amazing program this week. So we have been working on genetic medicine. And genetic medicine is very exciting as a modality for treating disease. One large reason for that is that it's modular. So I put over here this picture of a rocket delivering a satellite into outer space. And this simple modularity of a vehicle delivering a payload to the right place to do the right thing is kind of the underlying concept behind genetic medicine. And so I got inspired by this when I was in high school. I got a chance to work in the gene therapy lab, and there they were developing viral vectors for treating brain cancer or for ovarian cancer. And it's there, working with the scientists and mentors, that I became very excited about the potential of developing genetic therapies. So gene therapy has advanced quite rapidly, and the payload technology in particular has very rapidly improved. But what is lacking is the ability to deliver these gene therapy tools to the right place and into the right cells. So in fact, if you go onto the FDA website and you look at clinical trials for treating genetic diseases or other diseases, you will find that they largely target three tissues: liver, eye, and also cells in the blood. So there are many other organs in the body, and one of the reasons why we don't see many clinical trials for these other tissues and organs is that we don't have good delivery systems. And so because of that limitation, we have been focusing on developing new delivery approaches that may one day help get these things into the right tissue beyond the cells in the blood, the eye, and also the liver. So two short stories today. The first one focuses on a group of proteins from the human genome that are once upon a time derived from viruses, and we can harness these to turn them into delivery tools. And then the second story are kind of analogous prokaryotic systems that also once upon a time derived from prokaryotic viruses, and we can harness these to be able to deliver proteins and even other payloads into cells. So let me tell you a bit about these human proteins. So when the human genome was first completed, researchers were analyzing many of the genes encoding the human genome. And so one computational study here by Campillos and colleagues, they looked at the human genes that are similar to the retroviral structural protein GAG. And what they found is by doing structural homology search, they found that there are about 80 or so different genes, and they grouped them into these different classes. They are grouped based on their evolutionary origin. And so, for example, these sushi families are derived from the sushi family of retrotransposons. And there are also the scan family and also the paraneoplastic family. You can see that they're very well conserved in the capsid domain of these genes. And the capsid domain is what allows them to oligomerize and then form a virus capsid structure. So it's very interesting, and these are 80 out of the 20,000 protein coding genes in the human genome. So back in 2018, a couple of really interesting studies, they looked at one of these proteins called ARC. And so ARC is the protein that's expressed in the brain. It's thought to be important for synaptic plasticity and also for memory formation. And what they found is that this ARC protein is derived from retroviral GAG proteins, and also, at least in Drosophila and also in mouse cells, is able to form virus-like structures. And moreover, in Drosophila, these particles package RNA, and they can deliver the RNA across the neuromuscular junction. So this was really interesting, and I read this and I thought maybe if there's a large family of these different proteins, maybe they serve different functions. They target different tissues and deliver nucleic acids to these different types of cells in the body in a natural endogenous way. So we began to study these different proteins. So one of the family of proteins is this PNMA family of proteins. And so 3 of my lab members, Vicky, Yugang, and also Ramya, they began to study what do these different proteins do. So here is the phylogenetic tree that kind of gives you a sense of the relationship of these different PNMA proteins. And what you find is that they also have this blue capsid domain highly conserved among these different genes. There are several PNMAs in the human genome. And so the first thing they did is that they put all these different genes into human cells. And they asked the question, do they form capsids? And do cells release these capsids into the extracellular space to facilitate intercellular transfer? And so many of these PNMA proteins are indeed capsid forming, and they're also released by the cells. And one of these PNMAs is special, and that's the gene PNMA2. So this protein, unlike the other PNMA proteins, they're secreted by cells as protein-only capsids. So they're retroviral-like particles. They don't have a lipid membrane encapsulating the capsid. And so here are EM images showing the capsids that are released by the cell. And you can see that they are just a protein capsid. They don't have a lipid membrane around them. All the other PNMAs are secreted with the lipid membrane encapsulating it. So in many ways, this is like an adeno-associated virus particle. AAV is protein only, and so this is also a protein only capsid. We then thought to see, do these capsids package anything? Are they packaging proteins or RNA or other types of metabolites? And so we extracted material from them, and we found out that they don't really package anything. And here are sort of some of the RNA analysis that we did. You can see that the cells express PNMA2 RNA, but then if you extract RNA from the PNMA2 particles that are secreted by cells, they don't have anything in them. So they're kind of empty capsids that are shed by cells. So that's kind of weird. So then we studied these proteins biochemically. It turns out that they behave very well. So here on the left side, this is capsids that are purified from recombinant E. coli expression. So if you express this in E. coli, you can get them purified from cells as sort of intact capsids. It turns out that this protein is biochemically very well behaved. If you remove salt, so if you put it into 0 millimolar salt buffer, you'll see that the capsids will fall apart. And so if you put it under EM, you no longer see capsids being formed. But then if you add salt and also calcium back into the buffer, they will self-assemble back into these capsid-like structures. So it's kind of a nice nano cage where you can purify them as capsids, you can remove salt to disassemble them, and you can add salt back in to put them back together, almost like a Lego system. So we thought if this is a well-behaved system that we can open up and then also close back down, maybe we can then put something into it. We wanted to package RNA because being able to deliver nucleic acids would be very useful. So we solved the structure of this capsid using cryo-EM, and we found that there are some interesting bits of information. This is a cross-section of the capsid. So you're looking at the inside of these capsids. Red is negative charge, and blue is positive charge. And what this shows is that inside is quite negatively charged. So that kind of explains why we don't see any nucleic acids inside, because DNA, RNA have a negatively charged backbone, and so the charge would repel them from being packaged inside. So we thought, okay, if we know that the charge on the inside is not compatible, maybe we can engineer this capsid so that we can change the interior surface. And so we did that. We took the wild-type PNMA2, we removed a small fragment on the carboxyl terminus that is negatively charged, and we replaced it with a positive charge residue, or positive charge tail. So this is from the CCMV plant virus and is positively charged. We found that when you did this, the wild type can assemble capsids pretty well, The engineered version of PNMA2 with a positively charged tail can also assemble into capsids. But these capsids are assembled in the presence of nucleic acids. So when you put RNA in there, they form capsid. If you don't put RNA in there, they don't form capsid. So this is really interesting because now we have a protein that will assemble into capsids in an RNA-dependent way. And so we thought this is cool. Maybe we can use it to package mRNA and then deliver into cells to do something interesting. So we did this where this is an experiment where we had a reporter cell line. The reporter cell line has a flox GFP. So normally the cell is not fluorescent. But if we can deliver Cre recombinase into the cell, it would flip the gene on, and then we will see green fluorescence. And so this is what we saw. So we packaged the Cre mRNA into an engineered PMA2 capsid. We added RNAs just to get rid of everything that's on the outside. And when you put these capsids onto cells, you can turn on the GFP fluorescence. So this showed that we can use it to then functionally deliver a mRNA into the cell. And then on the other hand, if you just transfect in the mRNA, you also get activation. So this shows that we are sort of getting the expected functional signal. So this is where we are now. We still need to do a lot of work. These capsids, just like other nanoparticles, they are endocytosed into cells, but they don't come out of the endosome at a very high efficiency. And so this is something that we're working on now to try to get a system to be able to become more efficient. So related to this problem of endosomal escape, we have been working on this other system, which brings me to tell you about the second story of prokaryotic injection systems. So Joe is the graduate student who has been working on this. So the previous system, PNMA2, binds to the cell surface, is endocytosed into the cell, and then it has to come out of the endosome in order to release the mRNA into the cytoplasm for subsequent translation. We thought this endosomal escape efficiency is a problem we need to solve. And so one of the ways to solve it might be to just bypass the endosomal sort of endocytosis process. If we can directly inject something through the cell membrane into the cytoplasm, maybe this would allow us to do things a little more efficiently. So anyway, so this is where we began to work on this other system called Photorhabdis virulence cassette. This is from the bacteria Photorhabdis. And inside the Photorhabdis, they have, over the course of evolution, co-opted phage tails to become injection systems. And these are similar to secretion systems that bacteria use. They dock onto a target cell's membrane. They then inject physically something through the cell membrane and unload it into cytoplasm. But most secretion systems are membrane-bound on bacterial cell membrane. So to use those, you have to inject bacterial cells, or at least liposomes, with these injection systems embedded in the lipid bilayer. But the Photorhabdis virulence cassette are actually secreted, free-floating phage tails that have protein payloads in them. So here is the cryo-EM structure solved by Zhang and colleagues. You can see that this is a long shaft that has a hollow core on the inside, and you can load protein payloads into this hollow core. And they have a very sharp spike, and this is what it uses to poke through the cell membrane to unload into the cell. And then there are also kind of tail fiber proteins that allow this system to dock onto a cell membrane, recognize it, and then engage the physical injection mechanism. So this is a schematic of how this works. You have a syringe with a payload loaded inside. The tail fiber will latch onto the cell membrane, and this will activate this spring-loaded mechanism to then compress and shove this spike protein through the cell membrane to unload into the cell. In nature, this is a system that is expressed by these pathogenic Photorhabdis bacteria, and then it delivers a toxin into the insect host, and the toxin will lyse all of the intestinal cells and then kill the host. so that more of this bacteria can come out of the host and colonize new hosts. This is how it kind of spreads in nature. If you look into the genome of these pathogenic bacteria, you will find a gene cluster that encodes all of the structural elements of this syringe. You will also find right next to it a different cluster of genes that encode the payload genes. And these are the toxins that they deliver into the cell. Each one of these toxin genes has a signal peptide on the N-terminus. And so this is how this protein gets loaded into these syringes. So photowrap this is a little hard to work with. So we transfer these into E. coli, thinking that in a more genetically modifiable host, we can work with the system more easily. And so we transferred the entire locus, so all of the structural genes, all of the toxin genes into E. coli. And we found that we can make these PVCs pretty efficiently using E. coli. And so these are EM images showing the syringes that are produced by E. coli cells. And so they look very much like what the native Photorhabdis bacteria can produce. And so now we can try to see if they're functional. So we put these onto insect cells. So these are insect cells, SF9. And then we use the PVC produced by E. coli to deliver a toxin into these cells to see, can we kill these cells? And so here's the wild type version. So you can see a lot of the cells have now died, and there are also more red cells which are stained with a live or death marker. And so if they're red, it means the cell is dead or extremely unhealthy and on the verge of dying. And you can see that if you delete the toxin, there's no toxicity. If you delete the tail fibers so that it doesn't recognize the cell, there's also really no toxicity. And if you don't load the payload, there's also no toxicity. So this shows that we can use E. coli to produce fully functional syringes. Now, the question is, can we retarget them? So rather than targeting insect cells, can we get it to work on human cells? So the way that these things may recognize the insect cell, we surmise is probably mediated by the tail fiber protein. So we thought maybe we can change the binding specificity of tail fiber to recognize human cells rather than insect cells. So we did a lot of work to engineer the tail fiber. And Joe, my graduate student, probably tried more than 100 different designs, and they all failed. So it was very depressing at that moment. So eventually, right before we were ready to give up, AlphaFold 2 came out. And so we thought, why not just give it one more try? So we put the sequence of this tail fiber gene into AlphaFold 2, and we got this predicted structure. And so it was very hard to solve this structure because the protein is very flexible. So AlphaFold 2 gave us this structure, and so we used it to try to design different ways to engineer the specificity. So what we ended up doing is that we made a cut right here and removed this domain that we thought is what's recognized in the insect cell membrane protein. And then instead, we replaced it with either a binding domain from the adenovirus 5 virus or a DARPin that's been engineered to recognize different proteins. So either a DARPin that recognizes a surface protein, EGFR, or a DARPin that recognizes a lysozyme, which is just not a membrane surface protein. So now we can produce new PVCs with these engineered tail fiber proteins. And so what we found is that we can now retarget them. So here we're working with a human cell line, A549. This is a lung cancer cell line. It's positive for EGFR, so there's EGFR on the surface. And also it is a reporter that can get turned on if CRISPR-Cas is delivering to these cells. By the way, they're also targetable by AD5. So you can see it when we load these PVCs with the new tail fiber protein and also with the CRISPR-Cas, the wild type insect cell recognizing tail fiber didn't give us any signal. And if you truncate off this binding domain, there's also no signal. Now, if you put the AD5 binding domain on, you can get GFP activation. So this shows that we can now recognize human cells based on this bacterial injection system. And then if you inactivate the binding domain, you don't get signal. And also the EGFR DARPin is able to deliver CRISPR-Cas successfully, but then a different DARPin that recognizes lysozyme doesn't really work. And so this showed that we can now reprogram the system to be able to recognize human cells rather than insect cells. We can also load different payloads. So we can load Cas9 to do gene editing. We can load the zinc finger deaminase to be able to do base editing. So you can do something more than just activating a reporter protein inside the human cells. And then we also try to see how reprogrammable the system is. So we now engineered tail fibers with different epitopes on it. So either HA tag or flag tag or EE immune tag, etc. We then also generate reporter cell lines that can recognize these different tags. So now on the y-axis are these different antibodies capable of recognizing different tags that are now expressed on the surface of the reporter cell line. And so now you can see that when there is a correspondence between the epitope that's on the tail fiber and also the protein that's on the surface of the cell, you can get good signal. But then if they don't match, like if you have flag on the tail fiber, but then anti-HA on the cell surface, you don't get as much signal. So this shows that you can get a pretty nice one-to-one correspondence to get specific targeting and also delivery. So this is how we can reprogram it. And you can deliver this into the mouse brain. You can inject it into the hippocampus. They will recognize cells in the hippocampus, neurons, and they will deliver recombinase and be able to activate reporters in the mouse brain. So this can also work in vivo. So this is all good, but this requires that we load protein. But we also want to load other things besides protein to make it more useful. And so we thought, how do we design the system to be able to do this? So we turned again to nature for inspiration. So it turns out that the type 6 secretion system has a different mechanism for loading things and injecting things. Rather than loading the payload inside of the delivery system, type 6 secretion system has the payload that's functionally linked to the spike protein. So rather than injecting something through the shaft, this will use the spike to literally drag the payload into the cell. So we thought, okay, why don't we try to engineer PBCs to use this mechanism? So we then designed fusions of a cargo protein. So this is whatever you want to deliver, whether it's recombinase or it's Cas9 or other types of therapeutic molecules. You can fuse it to this sharp spike. You can fuse 1 molecule or 2 molecules or even 4 molecules onto the spike. And so I thought this can allow us to then do other things, because rather than using protein, we can also chemically conjugate nucleic acids or even other small molecules onto this for delivery. So this is the basic idea. So we now, rather than using the shaft to load things, we directly link something onto this spike, and then we use the spike to literally drag it into the cell for delivery. So here is what we did with Cas9. So now we fused Cas9 onto this PVC10, which is the sharp spike protein. And you can see now with one of these sort of PVC needles, you can fuse it and tether Cas9 onto the tip of the spike protein. So this is very cool. So now the previous example where I showed we delivered Cas9, we only delivered the protein, and the guide RNA was provided separately from this injection system. In this example, we can now mix this with guide RNA, and the guide RNA will get bound by the Cas9 molecule. And so when this injects, it's injecting the entire RNP into the cell for function. And so this is that. So here now we're using these sort of spike tethered systems to directly drag Cas9 and guide RNA into the cell to be able to mediate editing. So this works. Another thing that we did is that we also wanted to make the reprogramming easier. So rather than engineering fusions of the tail fiber with different binding domains, we thought maybe we can make a more clickable system. So here we used either strip tag or snap tag or spike catcher system. We engineered these tail fibers with these sort of catchable binding moieties. And so now we can take antibodies or other types of binding ligands and be able to just sort of chemically conjugate them onto the tail fiber. And so when you do this, you can then change it to recognize all sorts of cells. And so here is one example where we used a spike catcher to connect a single chain antibody onto it. And with this, we can achieve pretty good targeting in the cell, and we can deliver a toxin into a cell very, very efficiently. And so this is where we are now. So this is kind of the second generation of the system. So we can now directly tether payloads onto the spike, and we can also have a very modular way of changing the specificity of the system by just clicking different binding domains onto the tail fiber. So we call this the SPEAR system. Actually, we just sometimes just try to come up with cool acronyms. I don't actually even remember what the SPEAR acronym stands for anymore. But anyway, the SPEAR is just easier to remember. Okay, so these are two examples of things that we borrow from nature. And so I always get inspired when I see plots like this. This is a plot showing how much natural diversity has been sequenced. And so these are genomic sequences from all sorts of bacterial diversity. And you can see it's growing exponentially over time. And so there's tons of really interesting systems here, and probably many of them can be harnessed to do useful things for either biotechnology or human therapeutic development or even for agriculture. So that's what we've been doing, but over the past few years, we have also started to diverge into a new area. And I'm excited to also share a little bit of this new work with you. And the central area is about engineering resilience. Resilience is the ability of a biological system to move back to equilibrium. So if a biological system is subjected to stress, is pushed away from equilibrium, and then resilience allows the system to come back to equilibrium. This is how we recover from illness. If we get infected by a virus, we can come back to health. If we don't sleep very well, we can sleep and then recover from that. And then if we go exercise, we can recover from the stress that we experience through that. So as we get older, resilience declines. And so something that's really important for extending health span and improving human health is to be able to restore and to increase resilience in the biological system. So one of the things we started to look at is how do we increase immunological resilience? So the immune system is very important. It protects us from all sorts of things. And so the way the human immune system works is that in the early days, it develops immunity and it becomes really strong. And then in adulthood, it's very useful for protecting us against all sorts of infections and illness. But then as we start to age, the immunity declines, and then eventually it fails, and we die from that. And so the idea is that what if we can prolong this period of homeostasis? So one of the organs that's really important for immunological development and also maintenance is the thymus. So the thymus is where T cells are trained and developed. And so if we can somehow make sure that the thymus functions properly for longer periods of time, we can potentially enhance immunological resilience. And so unfortunately, the thymus is one of these tissues that starts to degenerate very early on in life. So this is the thymus of a 3-month-old infant. By the time that person is 17 years old, the thymus has already degenerated quite a bit. And then by the time this person is old, the thymus is basically fully degenerated and is no longer providing the function a thymic tissue is supposed to perform. These blue cells are thymic epithelial cells, and these are the cells that are secreting important factors for immunological development. And so as the amount of blue cells sort of disappear, then we lose thymic function. So Mirko is a postdoc in the lab who's been profiling changes in thymic function. And so what he did is that he looked at adult thymus and also looked at an aging thymus. And what he found is that there are specific factors that are diminished over time. And so we thought maybe we can make the thymus work or make the system work better by reconstituting some of these diminishing thymic factors. And if we can do that, we can maybe reconstitute it through delivery into the liver. And the liver is an interesting organ to choose because just like the thymus, all of the blood supply flows through the liver. And so whatever factors blood cells are exposed to when they travel through the thymus, they would also see those factors when they travel through the liver. And so we thought we can use LNP to then deliver these factors transiently into the liver to reconstitute liver thymic function. So what we ended up doing is that we identified 3 of these factors, DLL1, FLT3L, and also IL7. And then by packaging the mRNA of these into LNP, we can inject it in vivo, and then the LNP will deliver these fairly efficiently into hepatocytes in the liver. And so hepatocytes will take them up, express the protein. Some of these are going to be secreted, and the others will get expressed on the cell surface for signaling purposes. So more or less, we are transiently reconstituting some of these signals in hepatocytes, hoping that when blood cells go through the liver, they will get exposed to them. And so it turned out that this actually works really sort of well. So here is the paradigm. We use LNP to deliver over the course of 3 weeks in a mouse. And so these 3 mRNA will then lead to the expression of these 3 proteins in mice, and then we can then test the function. So one of the hallmarks of the declining immune system is that vaccination responses go down because the T cells are just not working as well. And so what we did here is that after the treatment with these 3 factors, we then provide the ovalbumin vaccine. So this is just a peptide vaccine that we're introducing into the mouse. And then we can measure reactivity to this vaccine over after vaccination to see how well is the immune system responding to it. And so what we find is that once we treat the system with these 3 factors, we get a boost in the immunological response. So compared to the control, where we put the luciferase mRNA in, the 3 factors allowed us to then boost the immunological response in these mice. And then also in both the spleen and also in the blood, you can see an increase in the amount of T cells that are now reactive to this sort of vaccination. So then we try to see how well is this working. It turns out that you can measure the biological age of the immune system by checking how much vaccination response the system is exhibiting. So here on this curve here, we're plotting the immunological response of different age of immune system. And so you can see over time, as the mouse ages, the immunological response declines. And so there's a nice curve that kind of tells you where the biological age of the immune system falls. And so the red dots are old mice, about 1 year old, that are treated with a control luciferase mRNA. And you can see the vaccination response from these control-treated animals are more or less sort of in line with their actual biological age. But if we treat them with the DFI factors, we can see that the vaccination response jumped. And now it is similar to a mouse that is half of his age. So about sort of a 6 months rejuvenated in this immunological vaccination response. And so this is something that we're pretty excited about because we can now sort of improve the resilience of this immunological response in mice. In addition to vaccination response, treating with DFI also allows us to slow down tumor formation. And so here, using the same paradigm, we'll find that the tumor volume in a mouse, when treated with these factors to boost T cell response, also was much improved in this experiment. So we can basically dramatically slow down the growth of the tumor. And then this also translates into prolonged survival for these animals. So here you can see that with the control mRNA luciferase, basically all of the mouse will die before 3 weeks. Whereas if we treat with the thymic factors, about half of these animals continue to live when we provide them with immune checkpoint blockade therapy. So this shows that if we can boost immunological function, we can improve resilience in these animals. So because we're manipulating T cell function, one of the question is, are we also increasing the likelihood of autoimmunity? So then we tested using this non-obese diabetic mouse model, which spontaneously develops type 1 diabetes. We treated with these factors to see, are we increasing the probability of diabetic occurrence, or are we accelerating the emergence of diabetic phenotype? And so we find that we don't really see that. So comparing the control luciferase treatment with the blue thymic factor treatment, we don't see any change or no overt differences between these different treatment conditions. So it suggests that a transient treatment of these thymic factors in the mouse allows us to improve resilience, but doesn't dramatically alter autoimmunity within these mice. So anyway, so that's where we are now. We're quite interested in thinking about ways to be engineer at the systemic and also organismal level to be able to improve resilience. Ideally, we can improve resilience across many different tissues to then be able to improve health span and also longevity. So last but not least, just want to acknowledge my team, and also this is my team at MIT. We're supported by a number of different foundations and also the HHM and NIH. And then last but not least, I just want to acknowledge, just want to thank the organizer again for the opportunity to be here. And thank you again. It's really great to interact with everyone.
Thank you, Dr. Zhang.